this granular process does is, the way we operate, bacteria, instead of forming this flow, they form this compact. This is a real compact granule. You see, this is not fluffy. This looks like a stone. So what happens is, outside of this granule, this is same as flow, but it's way more dense and compact. And this takes 30 to 40 minutes to settle. It takes few seconds to settle, perhaps 5 to 20 seconds. It settles pretty fast because the retention time for settling is really small. You don't need big tank to separate biomass from treated wastewater. So that's where this is called granular sludge. And the other thing is, in same process where outside you're bubbling oxygen, so oxygen needing bacteria they grow on the outer surface, and then those who don't need oxygen, they can grow inside. So in single granule, you can have different zones: aerobic zone, anoxic zone, that means nitrate but no oxygen, and inside we can have anaerobic zone, which means no nitrate and no oxygen. So in single granular, you can have 3-4 different processes taking place, and it is compact. So this is innovation in sludge treatment. This is some of the pictures from my lab. You see how compact they are, and these are treating. You see, like almost 600 up to 600, 700 milligram per liter perchlorate, and almost like 1500, 1000 to 1250 to 1500 milligram nitrate. Such high concentration of nitrate and perchlorate are treated in this reactor system. It's very compact. They settle down, like this is like too high, 2 minutes. They settle down like in 30 seconds. So very efficient process, and they are very compact. What is our other challenge? Surface water eutrophication. When you see Bangalore, I heard that it used to be called Lake City, but no more, because all the lakes around Bangalore, they are now contaminated. You see contamination. This is from China, but they have done wonderful job in China addressing the surface water quality problem. And as I stated, they not only cause green, they cause hypoxia because sunlight cannot penetrate. So fish and other aquatic organisms, they don't get sunlight, they don't get oxygen, so they die, lack of oxygen. And then this green stuff also over time decays means they generate organic carbon. The organic carbon settles down in the bottom. The organic carbon is now that is nothing but BOD and COD that get oxidized, consumes all the oxygen from the water column, and then water column, it becomes oxygen free and causes further problem to flora and fauna and aquatic organisms and the whole food chain. And the third thing is, of course, among these what we call algae, a certain class is called blue-green algae or cyanobacteria. And among cyanobacteria, some cyanobacteria, not all, some cyanobacteria, they generate toxins. And if think about that, if some lake is water intake for drinking water, all the toxins are now going to drinking water treatment plants. So this is the problem, and this is what happens, because of hypoxia, anoxia, fish, aquatic organisms, they die, and then there are toxins also. Generally, phytoplankton, we call them phyto, means from light, plankton means all the plants, they grow. They grow because of excess nutrient. We are discharging excess nitrogen and phosphorus to this river, to different river bodies. Climate change has caused, because not only nutrient, because of climate change, more and more new species of cyanobacteria are growing. They're flourishing. We used to see algal bloom in some stream, for example, or in some lake system in July. Now we are start seeing in May, early May. So their duration has increased. They used to be maybe 10 days, 15 days. Now they are lasting for 2-3 months, which is bad scenario. So all these challenges happens. And then what causes HAB? As I stated, overgrowth of algal species, nutrient loading, and these nutrients are coming from municipal discharges, agriculture runoff, urban runoff, industries and homes. And one of the main consequences are, of course, these toxins, what we call cyanotoxins, and they can cause these different symptoms. They can cause liver failure, they can cause skin problems, they can digestive problem. Neurotoxins can cause nervous system and other related problems. And these are all real Limitations, how do you identify? The most common tool is take a sample and look into microscope, and then morphology is defined. Based on morphology, people say, oh, this is this type of cyanobacteria, this type of algae, this is this type of cyanobacteria. But sometime two different genera, they can look alike, same in under microscope, but they're different. So microscope has its own, still it is widespread use technique, but microscope has its own limitations. So what we do is genetic tools. So we extract DNA, and then we can sequence, and then after sequencing, we can do bioinformatics, and we can identify what genera, what taxonomic classification is. And this is some real data from my lab. We can do this sequencing 2016, genus level, these all cyanobacteria, see Cenococcus, Femisomenon, that how it changes from June to July to August. You see the taxonomic classification there, then their relative abundance changing at different sites and different months. And then this is 2017, likewise from same lake system, phylum level, and then at genus level, they're changing how at different site in same lake, but you see the taxonomic classification and diversity. At one site, you have more of this Cenococcus. At the other side, you have more of this green, which is Femisomenon flock aqueous. So these things can give you genetic information, can give you nice information about how, what we can do, what kind of cyanobacteria are growing, and what can we do about those. Because once we know their taxonomic classification, we can find out their nutrient dependency, and from there we can develop lake management system. So I think I have I'll take some more questions. These are challenges of our 21st century. I will end on a suggestive note that we cannot just sit and take a backseat as a citizen, as the community member. It's our duty to do something and then play our own role. So this is all I have today. Thank you so much, and I will be interested in taking any questions.
Thank you very much.
Thank you so much for this wonderful talk. But still, there may be few queries among the young minds. So therefore, I would like to request Professor and Dr. Durga Madhav Mahapatra to address this question and answer session. Dr. Durga Madhav, please.
Thank you very much, ma'am. In fact, it was a really interesting and a very thought-provoking talk by Professor Goel. And as he went on to highlight some of the key challenges in 21st century, we would now like to open the floor for any discussions and question answers. We will take up queries from students, faculties, the dignitaries, and they might kindly raise their hand or else post the questions in the chat box so that we can take them, and Professor Goel would be able to respond to it immediately.
Sure.
If there are any questions, kindly raise your hands. We have already a few questions in the chat box. In the interest of time, I would like to read some of them.
Sure.
This question is from Abhishek Behra. His question is, how lab culture microbes are enough to compete against various microbial species in an uncontrolled environment?
So, Abhishek, did you mean lab cultures for different contaminants or different lab species? If I understand, I think different lab cultures, how they can compete for different contaminants. They have specific, as we know from the rule of microbiology, so far we just know about 1% of microbes present on Earth, only 1%. That means we don't even know about 99%. And we don't know about 99% because we don't know how to culture them. We don't know how to isolate them. And isolation techniques are really biased. And some of those which are there, of course, you have to do really a lot of effort. So it depends upon under what environment, you're isolating those cultures, and if you throw them to unknown environment, they're less likely that they will cope up that pressure. But more and more bacteria are coping up pressure. They have special tools to cope up this external pressure. So depends upon how versatile your culture is. Sometimes they survive, but if you throw them to unknown environment, they may not survive.
Thank you so much, sir. There's another question from Ashwini Kumar Sahu, and uh, he would like to ask, how is the genomic sequence useful for finding the taxonomic position of the species?
Yeah, genomic sequence depends upon if you want taxonomic classification, what we do or around the what we call metagenomics. And metagenomics you can do at 16S label, that means 16S rRNA gene. You can sequence whole. That used to be the trend. Now people do amplicon sequencing. You get millions of read all together of 16S, and then you can do taxonomy because it's very conserved. 16S is very conserved, so people still use 16S for taxonomic classification. That means it tells that, okay, who is. I can give you analogy. For example, in a room there are 20 people, so the 16S can tell you, okay, this who those are. Well, there is Dr. Mahapatra in the room. There is Kabir Sethi, there is Dr. Goyal, right? Can tell you, but the tool cannot tell you what Dr. Mahapatra is doing there, what his characteristics are. That may display that, okay, he's there, that's it. But what he does, under what circumstances he reacted, under what circumstances Dr. Goyal reacted, that you can do with more advanced genomics, what we call the metagenomes, where you sequence the whole DNA and then you assemble them. In parallel, you can also sequence RNA also, that means gene expression, and that tells you their ecophysiology, that what kind of function they are doing. So it depends upon what level of analysis you want to do. But in short, yes, the sequencing can tell a whole lot of things about the microbial diversity, their abundance, and their ecophysiology.
Thank you, sir. There's another question from Professor A.B. Das, who is the CSIR Emeritus Professor with us at the Department of Botany. He would like to ask what percentage of awareness is achieved in environmental awareness in rural and urban sector globally so far, whether global leaders put their efforts in grassroots level to effectively mitigate environmental issues.
Difficult to say what percentage. We need more. There's more and more awareness. The thing is, what is killing us is this consumerism, right? Example I can give you is, I mean, I used to do neem datun. I hail from village, okay, but now if you go to even simple mart, right, there are 50 different variety of toothpaste. Why do you need 50 different variety? Because there's competition, and and if you go to a simple shop, if you have one or two variety of toothpaste, people don't go there. You know what? People say, oh, we don't have options there, right? They don't go, and this is consumerism. So so that is one problem in the environment. About the leadership, there are awareness, lot of awareness, but again, it's all about like politics versus leadership, right? In the environmental awareness, what we do prefer. uh, I would say yes. Still Europe and US, these countries are well ahead of many countries. In India, we have way to go. We have a lot more to do in terms of environmental awareness.
Thank you very much, sir. There's another question from one of our research scholars from the department, Karishma Das, and she would like to ask how microbial interaction is helping in wastewater treatment. I mean to say, what might be the association among the various, let's say, communities in the wastewater?
The thing is, as long as for engineer, that's what we don't care about microbiology. That was the slogan before. I would say before 2000. But then more and more engineer, like for example, I have never taken a single course, biology course in my life after 10th grade. But 80% of my research is microbiology related. And the thing is, it helps so much. So when an engineered system is working fine, especially bioengineered system, engineers don't care about microbiology. But then suddenly some upset takes place, something happens, and you monitor different parameters. Well, my influent rate is correct, the temperature is correct, the pH is good. Why the system is not working? And that's where the microbiology helps. Like you look into microbes and then say, well, oh, look at this particular substance has caused toxicity to this microbial community. I'm expecting ammonium oxidation, but my nitrifiers are not there in the reactor system, right? And that's where it helps. This is just one simple example. And of course, how can you enrich? How can you manipulate microbial community? How can you make bioprocesses more sustainable, more, and that's where metabolic pathways, really helps. I mean, another typical example is when you do genomics. Um, for example, let me give you an analogy. Like, for example, Dr. Mahapatra and myself, we work in a kitchen, both of us, right? And then somebody comes and outside order food, and they get nice chapati. Okay, so for the person eating chapati, all he cares about is chapati coming, and he eats. But then inside the kitchen, what is going on is I am making the dough, and Dr. Mahapatra is making chapati. If I don't make the dough, he cannot make chapati. So now convert this in terms of microbial ecology, and that is exactly what happened. So if I disappear, you won't be getting chapati. And that exactly happens between microbes. There are many microbes, they synergize their effort. A is helping B, B is helping C. And you wouldn't know from engineering tool, you have to use genomic tools to know that.
Thank you very much for a very lucid and a very interesting answer from you, sir. We can just take up one more question, which is from Karisma again, and she would like to uh, ask whether the uh, there is any difference between the treatments for the surface and groundwater.
Depends upon source. There is, of course, treatment. Surface water, you need to treat a lot more, a higher degree than groundwater. But again, if groundwater is contaminated, you need same degree of treatment. But I remember, in villages and in many parts of even here in Utah, actually, I live in US. Uh, People just directly take groundwater and drink it. No treatment is needed. So if it is pristine clean, you don't need actually. All you need is disinfection perhaps. So yes, the degree of treatment are different for both,
Thank
but
you very
depends
much,
upon.
sir. Thank you very much, sir. And in the interest of time, we will restrict the questions here. And if there are any further queries, we might have a chance to again interact with sir, maybe when we host him again in the future. And
Sure.
now I would like to again invite our advisor, Professor uh, 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 Bhavtarini ma'am, to take over and follow it up.
Thank you, Durga. Thank you so much for coordinating.
Thank you, sir.
Appreciate it.
Thank you, Professor Goel. And thank you, Dr. Durga Madhav Mahapatra, for nicely coordinating the question and answer session. Now, I would like to request Professor Prabhati Kumari Mahapatra, advisor and coordinator of this COE, to give her concluding remark. Madam, please.
Thank you, Bhavtarini. Am I audible?
Yes.
Yes, madam.
Okay. So a very good morning and of course, good evening, Professor Goel. Honorable distinguished keynote speaker of today's international webinar, Professor Ramesh Goel, Professor Kabir Mohan Sethi, coordinator of the center, convener of this international webinar, Dr. Bhavtarini Panda, co-convener, Dr. Durga Madhav Mahapatra, esteemed members of organizing committee, esteemed faculty members of Utkal University, outside the university and colleges, dear student participants. I feel honored to be part of this international webinar. At the outset, let me extend my heartfelt thanks to today's keynote speaker, Professor Ramesh Goyal, for accepting our invitation and delivering a very, very wonderful and very informative talk on environmental engineering in 21st century, what we have learned so far and what we can do despite the difference in the time zone. Thank you very much, sir. In fact, Professor Goel has covered very, very important aspects like advances in environmental engineering between 1900 to 2000, environmental regulations between the same time period. Then he has focused on several public health concerns like having sustainable food, water, and energy supply. And he has focused on establishing efficient, healthy, and resilient cities as there is migration of rural population to the urban areas. Then he has focused on key issues the mankind facing globally, like water scarcity, condition of the soil, and diminishing non-renewable resources. Dr. Goel has also emphasized on various causes of water pollution with suitable examples, including the Chilika Lake, and he has focused on the misuse of water, like consumption of water, contamination, and affluence present in the and our lifestyle, the present lifestyle that is a matter of real concern. Then the occurrence of microplastic, which is the recent finding, and this microplastic in tap water globally is also very, very important, and we need to take care of that particular aspect. Then the vanishing water bodies, that as he has aptly given the example of his own village, that how the wonderful and people used to depend on the water bodies now have disappeared due to dumping of plastics. Then next one, that is the superbugs, the antibiotic resistant bacteria contaminating potable water globally. So even our country is not spared. So the moving from simplicity to modernity is the challenge of the years to come for the whole mankind. So that has to be taken care of. So need of the hour is how to recover natural resources, then reducing carbon footprint, and most challenging, removal of contaminants, including micropollutants, micro and nanoparticles, then polyfluorinated alkyl substances, and the most important or the matter of concern is pathogens, about whose biology everything is not understood. So let me thank Professor Goel once again for his very, very informative and of course, as told by Dr. Durga Madhav, that thought-provoking. The youngsters who have joined must have learned and enjoyed Professor Goel's talk. And the last part was the question answer, which was very, very, properly the questions were answered. And then let me extend my heartfelt thanks to our Honorable Vice Chancellor, Professor Sabita Acharya, for her support and advice in conducting this webinar. My thanks to our Chairman, PG Council, Professor Basant Kumar Malik, and Coordinator RUSA, Professor Prabodh Kumar Hota, for their encouragement and advice. My sincere thanks to Professor Jaggeswar Danpat, one of the members of the committee, and for his valuable suggestions in smooth conduct of this webinar. Thank you Thanks to Dr. Josna Rani Pradhan, Dr. Chinmay Pradhan, Dr. Kanhu Charan Satpathy for their valuable inputs as committee members of this webinar. My sincere thanks to Professor Kabir Mohan Sethi for his welcome address. I also thank Dr. Bhabatarini Panda, convener of this webinar, and Dr. Durga Madhav Mahapatra, the co-convener of this webinar, for conducting is very smoothly. Last but not the least, I would like to thank faculty members, research scholars, and students for their active participation, and finally making this webinar a success. Thank you.
Thank you, madam. So I think we have arrived at the end of the webinar. And thank you all for participating in the webinar. Thank you.
Thank you.
So
Thank you very
we can
much.
Thank you, one and all. Thank you so much. Thank you very much, sir. Thank you all. Thank you very much. Yeah. A happy New Year in advance to all.
Yes, Professor Goel. We also extend our heartfelt thanks as well as our best wishes for a very happy new year to come.
Thank you so
Thank you
much.
very much, Professor Goel. We extend our heartfelt thanks to you, and also a happy Christmas over there, and also happy New Year. Thank you very much.
To you all, thank you so much.
Thank
Namaste.
you very much. Namaste.
So we
Namaste.
welcome you sometime to our institute, and whenever you feel like, sir, just inform us and we'll be there to host you.
Yeah, my former student is at CISIR Bhuneshwar, Tripti Das. I don't know if you know. Dr. Tripti Das. So I come there, I gave talks. So I may come in March sometime. So
Thank you so much, sir, and we'll
Please do
let
that.
you know.
thank you so much.
Thank you.
Thank you.
Thank you so much, sir.